Assalamualaikum warahmatullahi wabarakatuh and salam sejahtera. I'm Madam Zabaria binti Muhammad Jusso. I'm going to teach you biology and today we're going to learn about chapter 7, recombinant DNA technology. Introductions to recombinant DNA technology. Our learning outcomes are: define recombinant DNA technology, list the tools used in recombinant DNA technology, that including the target gene, the gene of interest, restriction enzymes, DNA cloning vector, host cell, and modifying enzyme. What is recombinant DNA technology? Recombinant DNA technology can be defined as the technique involved in altering the characteristic of an organism by inserting genes from another organism into its DNA. Tools used in recombinant DNA technology are target DNA or gene of interest, restriction enzyme, DNA cloning vector, the host cells, and the last one is modifying enzyme. What is target DNA or target gene? Target gene can be defined as DNA fragment containing gene of interest to be cloned. The second tool is restriction enzyme. Restriction enzyme can be defined as an enzyme that can cleave or cut DNA molecules at restriction sites. There are many bacteria that can produce the restriction enzyme. Example of restriction enzyme is EcoR1. EcoR1 recognizes the sequence of GAA TTC, and it will cut the sequence within the G and A to produce the sticky end. The second example of restriction enzyme is SMA1. SMA1 recognizes the sequence of CCC, GGG, and it will cut the sequence within the C and G to produce the blunt end. The functions of restriction enzyme is to recognize and cut or cleave DNA at restriction site by breaks down the phosphodiester bond within the DNA sequence to produce sticky ends or blunt end. The same specific restriction enzyme will be used to cut open the bacterial plasmid and to cut out the target gene at their respective restriction sites. What is restriction site? Restriction site contain palindromic sequence. Palindromic means a words, phrase, or sequence that will sound same if you read it forward and in opposite way. What do you notice about these phrases? The first one, radar. If we read it in opposite way, it will sound radar. Let's try the second one, race car. And it will sound same in opposite way, race car. Okay, this is the third one, madam, I am Adam. Madam, I am Adam. Okay, student, you may try the rest by yourself. Okay, the third tool that you use in this process is cloning vector. Cloning vector can be defined as an agent or small DNA molecule that carry the gene of interest, and they will combine to produce the recombinant DNA, then they will be put into the host cell during the genetic engineering. Cloning vector consists two or more restriction sites. They also have origin of replication and also a selection marker genes for screening, example ampicillin resistant gene. The host cell is defined as the cell that can receive the recombinant DNA for the cloning purposes. And the last tools used is modifying enzyme. Modifying enzyme can be defined as enzymes involved in genetic engineering to degradation, synthesis, and alterations of the nucleic acid. The example of modifying enzyme is DNA ligase. How does the DNA ligase work? As we know before, the plasmid been cut open by restriction enzyme, and gene of interest also been cut out by the specific same restriction enzyme. These both then will be combined together by DNA ligase. DNA ligase will ligate them together by using the phosphodiester bond, and this will form the recombinant DNA plasmid. So student, you may try this question. So that's all for today. See you next time. Thank you.